last part of this lab, we'll look at karyotyping and cytogenetics. Cytogeneticists are scientists that examine chromosome structure, seek out the causes of chromosomal abnormalities, and study how chromosome structure relates to an individual's phenotype, their appearance, and biochemical traits. Abnormalities in chromosomes can result in early embryonic death, congenital defects, the development of cancer, and infertility or sterility. Studies of chromosomes begin with the extraction of chromosomes from cells. The chromosomes are then placed on a glass slide, stained with dye, and examined under a microscope. Each chromosome pair is assigned a number from 1 to 22, and then XY that is based on staining pattern and length. A karyotype is a representation of a person's chromosomes. The chromosomes are shown in pairs and arranged in order of decreasing size. Individual genes cannot be seen on chromosomes. What to look for in a human karyotype? First, the number of chromosomes. Are there 46 chromosomes? And then with each chromosome pair, is there one pair of each autosome and one pair of each sex chromosome? Are there any deletions, rearrangements, translocations, or other abnormalities in the chromosomes? Karyotypes are presented in a standard form. First, the total number of chromosomes is given, followed by a comma, then the sex chromosome constitution. If the chromosome or abnormality represents an addition or deletion of a chromosome, that is then indicated after a comma beyond the designation for the sex chromosomes. Here's a list of some common genetic disorders that occur due to chromosomal abnormalities. In Down syndrome, an extra 21st chromosome is present. Turner syndrome is where a sex chromosome is missing, so that the individual has a total of 45 chromosomes. Klinefelter syndrome is a syndrome found in males who have 2 X chromosomes. Remember that it's the Y chromosome that determines the sex of the individual. Having one copy of the Y chromosome makes an individual a Y, regardless of how many X chromosomes they may have. Here are some additional terms that you will need to complete the rest of this lab. In a monosomy, this represents the lack of one of the pair of chromosomes. A common monosomy is X chromosome monosomy, known in humans as Turner syndrome, and designated as 45X. Conditions of having 3 chromosomes of a particular type is called a trisomy. A common trisomy in humans is Down syndrome, or trisomy 21. A male with Down syndrome is designated as 47 XY 21 plus. Some additional type of chromosome abnormalities are due to changes that occur not in the total number of chromosomes, but within the composition of the individual chromosomes itself. A deletion occurs when a chromosome piece breaks off and is lost. A translocation occurs when a piece of a chromosome breaks off and attaches itself to another chromosome. The condition in which a man or woman has difficulty conceiving a child is called infertility. Infertility can result from chromosomal abnormalities. The technique in which fetal cells are obtained from the fluid surrounding the fetus, or amniotic fluid, to be used in genetic analysis is called amniocentesis. There exists a group of cancers involving the abnormal growth of white blood cells. This is called leukemia. Leukemia can result from chromosomal abnormalities. Failure chromosome pairs to properly segregate during sperm or egg formation can result in non-disjunction. During meiosis, or the formation of sperm and egg cells, 4 haploid cells should result. Haploid cells are cells that contain a total of 23 chromosomes. If in any of the 2 main stages in this process non-disjunction occurs, sperm or egg cells are going to be generated that either have an additional chromosome or a shorter chromosome. Most chromosomal abnormalities of the autosomes, pairs 1 through 22, involving the absence of a chromosome, do not result in a viable fetus.